I will introduce Nadina, who is one of the most passionate individuals about RNA. So Nadina, if you come on up, turn it to you. Just push the arrow.
Well, good morning, everybody. I'm Nadina. I'm extremely excited to be here with you today. So what I will do is I will take you through the journey of how we create optimal ASOs in the n-Lorem lab. So essentially, as Sarah mentioned, I wear two hats. I'm leading the ASO design and discovery in the lab, but I'm also overseeing all our research efforts, both directed research and also basic research, in order to advance the technology with the ultimate goal to serve more and better the nano-rare community. So this is exactly that. So the umbrella and the most important thing that I would like you to remember for the entity of this talk is essentially the reason that we are able to do what we are doing at n-Lorem every single day is we're really standing in the shoulders of giants. So we really benefit and leverage more than 35 years of ASO experience, and really Stan and all of his colleagues at Ionis, we are leveraging all of this experience and expertise. And at the same time, what we're doing, leverage on that, we want to build on continuous research innovation. So research innovation is something that you need to understand very well what is already known and try and make it even better. And this is how transformations happen. And this is exactly what the ultimate goal is. My team's goal is really to assure that each patient is treated with the best ASO possible. And I would like to take a step back and just introduce a little bit about these beautiful molecules that are called ASOs. So what are ASOs? What are antisense oligonucleotides? And I'm sure you've all heard historically the really big players in drug discovery. And these are small molecules and monoclonal antibodies. And even though, as I said, they are big players and they've been around for so long and for decades, actually their rules of engagement and exactly how they work remain elusive and not very well understood. So we don't know exactly how they engage with their receptors. Now, on the opposite end of the spectrum, we have these beautiful molecules. We have ASOs, where very interestingly, their receptor is actually an RNA molecule. So what this, and also importantly, what we know is we understand very well the rules of engagement. Everything makes sense. And we leverage exactly that. So, and essentially what happens is we have an ASO and finds its target mRNA by simple rules of hybridization. It base pairs, it clicks together. So you can imagine that you have an ASO, finds the RNA in the C of the cell, and like a zipper, it binds it and it targets it. And when it targets it, it essentially modulates its gene expression. And this is exactly what we do in order to treat and create ASOs. So it is these properties of the ASOs, as I said, that enable us really to treat this community and to do what we do at n-Lorem in a high quality, high throughput, and cost-effective fashion. For example, if I were to do a face-to-face comparison with small molecules and monoclonal antibodies, as I said, the rules of engagements are either remain elusive still or are partially understood, whereas for the ASOs, easy to use, we know exactly what will happen, and they're very well understood. Because of this reason, we are able to learn from all our past experience, and we can actually transfer this knowledge into the next patient and into the next patient and become better and better and better. And this is really the power of this community. So broad transferability, whereas in a case of a small molecule, for example, you may have all heard the phrase, change the method, change the drug. So the cost of drug discovery is cost effective, so it's much more modest than what it is for other drug modalities. And importantly, and I will show you how we do that, the process of the drug discovery takes months. It is very much accelerated. It does not take decades or years the way it does for other drug modalities. Now, because of the ASO technology and because of the mechanisms that we will go in a moment, the versatility and the specificity are very high. ASOs, depending on their design, can act in very specific ways in mechanisms that we completely understand. So therefore, before a case enters, and I will go through that, we need to see, okay, how ASOs can therapeutically intervene with this disease, and what is the ASO strategy that we need to assess. And then finally, and this is also exactly what we're doing, we are able to advance the technology. Because it is well understood, we are able to make it better in an extensive manner and rapidly. And this is exactly what we do. So what do we do exactly in the n-Lorem lab, and how do we do it? So the research that my team does at the n-Lorem lab has 3 phases. The very first thing that we do is we do ASO discovery. So we essentially we design and screen ASOs in order to create that optimal ASO that eventually will become the clinical candidate. Then the other thing that we do is we do research activities, and we have very specific reasons of why we're doing them. We have directed research programs, and we do this in order to better understand targets and their mutations. And this is very important in order then to understand what kind of ASO we need to design, and if ASO technology can help. And finally, we do basic research in order to advance ASO technology. What we want to do is we want to create more efficient, safer ASOs and reach more patients faster today. And this is the reasons that we are doing these other research activities, and specifically basic research. And I will take you through some of these examples. To achieve all of this, what we did was we purposely created a lab and a team with the right skills, right experience to really meet the demand in the highest quality possible. And this is the n-Lorem lab. Now, if you want to create a team at n-Lorem that is outstanding, because our mission is just so wonderful and everything that we do is to serve you, we really need to have a scientific team that is extremely robust with really the right skills, the right ingredients. Because if you have that, then n equals 2 does not equal 4, it equals 5 or even more. What are these ingredients? And what you will start seeing here is you will start seeing the names of the people that comprise my team, and always you will see Stan's name on it, because of course we are in constant communication every day, and he oversees also all of these activities. So just to go on the ingredients, what do we need? So first of all, we need RNA expertise. Why do we need RNA expertise? Well, RNA is almost the beginning and the end of what we do. It's really our target. This is what we're targeting. We're targeting mRNA. RNA is our medicine. And it's also really this beautiful molecule that drives health and disease. So we need to be able to understand it, tame it, and really use in our advantage its beautiful properties. We also need to know and understand and have expertise on cell and molecular biology. We need to be able to understand the phenotype of the cell, of what the disease causes, to ask the question, can an ASO really correct this phenotype? Can we really therapeutically intervene with an ASO? The other part, of course, is ASO's mechanism of action. We need to understand how ASOs act. And the reason we also need to understand how they act is we need to know exactly what it is we need to do for each patient. This is really a personalized medicine, and how we can improve it even more. Of course, we need ASO design and discovery skills, because obviously this is really the core of what we need to do in order to identify this optimal ASO. Drug discovery and automation requires a very specific skill set, and of course this is something that we also require to have. And definitely, last but not least, and I will go into further details in a moment, we also, of course, need to have a very robust bioinformatics capabilities. And this is actually something that we built in-house, because we need to have artificial intelligence, AI-driven ASI design and discovery, and leverage all that. So all of these ingredients then, we have created all of these ingredients together in the n-Lorem research team. And it's these skills and experience that come together and mean that we can provide the best possible decisions for all of you. So how did we start? Well, we actually have come a long way. And we started as a very small lab that we funded at Ionis, but of course the demand was very high, so we needed a very robust capacity. We needed our own capacity. So today I'm extremely proud and very, very excited to show to all of you what we have accomplished and how far we've come. We've essentially done a very rapid progress with a very newly established lab with all of the people that I showed you before. And here on this diagram, you can see essentially how many discovery programs are going through our lab, our scientists do, every quarter. We started in the n-Lorem lab as 0 in 2022 because what we had was actually the portion at Ionis. And you can see this very sharp increase where we right now have, more than 8 programs per quarter, and we continuously increasing. I mean, this is just the beginning. The sky is the limit. Who is responsible for this, and who is responsible for this progress? These faces there are the faces of the names that I showed you just a moment ago, and these are all the scientists in the n-Lorem lab. And these are really the people that make the magic happen together with the machines, together with robots, right? So this is really a match made in heaven. So you have the people at the top, you have the machines at the bottom. The machines are the ones that allow really high throughput screening and allow very rapid, high throughput, robust, in a very high quality manner screening. This is how we're able to do that. So the conclusion from this part is that I'm very excited to tell you today that in the less than 2 years, we now have more than 27 programs that our scientists, the small team that you saw there, have completed discovery, and they're progressing into preclinical development. And we are really at a very fast rate with, importantly, with very high quality data of having 8 programs and more per quarter, and we are really continuing to grow. And as I said, really, the sky's the limit. We definitely have the capacity to do even more. We can really double or triple the next year if we raise the funds. Now, I showed you what we do at the n-Lorem lab. As I said, we have research activities, and we also have, of course, the ASO design and discovery. I would like to take you through this process and break it down. How do we go from a patient submits an application to actually go and handing in an ASO that we think is optimal? So we have several checkpoints, and this is where, let's say, my teams and my responsibilities begin. So the very first is a case is submitted at n-Lorem, and as Sora said, we have a panel of experts in its particular case, and I will show you where they essentially review the case. My team's responsibility is to really assess whether ASO technology can meet the needs of this particular patient. If the answer is yes, and if all of this committee we agree and recommend that the case is accepted, then we move into ASO design. We design these ASOs, and then we move them to discovery. We move them to discovery to screen. After discovery finishes, we ask ourselves the most important question of all. Did we find optimal ASOs? If the answer is yes, then we move to rodent tolerability studies, in vivo studies. If the answer is no, then we think of 2 things. Did we do every single thing that we could to identify the ASO? Can we redesign? Can we start over? If the answer is yes, we go ahead and do that. If the answer is no, then we would need to discontinue the case because we did everything that we could. Whichever the answer is, what essentially my, let's say, kind of responsibility finishes on where we present at RMC. So essentially there we take forward a lead candidate where we recommend based on all the activity and discovery data, or if there is a challenging case that we think that we recommend that it should be discontinued, we bring to this committee as well, which is something that both Stan and Sarah touched on. Right, so I would like now to take you kind of step by step on this process. So the ASO review, how you submit, your physician submits an application, what goes through at least my mind to see if the ASO technology can meet the needs of the patient. The very first question that I need to ask and understand is the mutation amenable to an ASO? And more importantly, do we know enough about the nature of mutation to really say that the ASO is the optimal therapeutic intervention? Then the other thing that we need to understand is, do we understand enough about the proximal pathological molecular mechanism? This is very important because essentially in order to intervene therapeutically, we need to be able to correct these phenotypes with an ASO. So when we know the phenotypes, then we can go and ask the question, can we correct? And very importantly, sometimes we do not know these answers, and depending on the situation, perhaps we go ahead and do further research. And there are a lot of times, as I will show you in a moment, that we actually add into new insights into the pathology of disease. Now, lastly, what is the ASO design strategy? What do we need this ASO to do? What exactly is the optimal therapeutic intervention for the particular patient? Do we need an allele selective ASO? And I'm sure you might have heard that before. What does this mean? An allele selective ASO is an ASO that specifically reduces the abnormal mRNA while leaving the normal, the wild type mRNA untouched. And I will show you how we designed that. Do we need that? Or are we okay? Is the protein not essential? And are we okay in actually reducing both the abnormal mRNA, and therefore the abnormal protein, and the normal one? Or do we actually need to modulate somehow this mRNA? Do we need to change its splicing? Can we use a steric blocking ASO? All of these are questions that we need to understand and we need to know before a case enters design. Is there mutations that we cannot fix? The answer is yes. True null mutations, when you have a true loss of function, either protein not being produced or not completely active, these are mutations that we cannot fix. Now, and this is really a real-life slide of the committee that Stan and Sarah talked about. This is the ATTC committee. And essentially, this is a committee where a group of experts come together, review the application, and based on their specific expertise, we essentially make their recommendation of accepting or declining a case. And you can see this is a real-life slide, so you can see all the different expertises here: clinical genetics, ASO design and discovery, clinical development, clinical operations. In this particular case, and that was something of my recommendation recently, for this particular program, for example, that was amenable to RNase H1 ASO, and I was recommending there that for now, based on what we know, we need to have an allele selective ASO. Right, so in this particular case, then we have something that a case is accepted, so then it really enters the n-Lorem lab. So then we need to design ASOs. What do we do? The first thing that we need to answer is, what do we need to do to this mRNA in order to treat? What we need to do to this mRNA, it will depend on what kind of ASO we will utilize in order of what kind of strategy of mechanism of action this ASO will do. The two classes of ASOs are essentially the gapmer. Perhaps this is the most, I don't know, maybe famous one, where essentially you can see, and this is, you see this really beautiful molecule where you have a combination of RNA bases and DNA. So you have RNA bases at the wings, so at the very left and at the very right of what we're calling, and you have DNA bases, 10 DNA bases at the very center. Now, the center is where this magic happens. The center is essentially what creates the substrate for one of the best enzymes in the world, RNase H1, that goes and cleaves the RNA on that hybrid. So when the ASO binds the RNA, the DNA part is what hybridizes exactly, all of it hybridizes, but this is where exactly the RNase H1 goes and cleaves. And as you can see here, I'm not going to point, the target RNA gets degraded. Now, very importantly, ASOs get chemically modified. Why? Why do we chemically modify them? Well, it has been shown, and this is all the research that was done also at Ionis, was that essentially we need to have specific chemical modifications in order to improve the pharmacological properties. And for example, we have the 2' methoxyethyl modification in the RNA, where we know that it can improve the affinity to the RNA target, reduce immunogenicity, and extend the longer duration of action. The other modification that we have is also phosphorothioate that is on the backbone on the ASO, which improves the stability and interactions with other proteins, and all in all improves the pharmacological properties. So the gapmer ASO is something that we use when we want to reduce an mRNA target. Now, when we want to reduce an mRNA target in an allele selective way, as I was telling you before, what we look for in a gene is SNPs. And this is something that Stan introduced yesterday in their talk, and I'm sure you've all heard the word. What are SNPs? SNPs are single nucleotide polymorphism. They're essentially spots. They're essentially just differences in the mRNA of the abnormal mRNA, the mutant mRNA, and the wild type normal mRNA. So what we do, and this is exactly why we need the whole genome sequencing, a very crucial part, is we go really and read the entire gene, and we are looking for these little differences. These little differences are benign, they don't cause any pathology, but they help us to tell the RNase H1, that enzyme, hey, come here, cleave this abnormal mRNA, but leave that one untouched. Right, so that is the first part. Now, the second class of ASOs is essentially steric blocking ASOs. Here, you don't have an enzyme that goes and cleaves. Here, as its name says, it just acts by occupancy. Perhaps the most famous that you may have heard are splicing ASOs. But these ASOs differ from the gapmers because, as you can see, these are all RNA. And again, same modifications, methoxyethyl, the 2' and also the phosphothyroid linkage throughout. And usually this is an 18-mer. And essentially, you put these ASOs depending, you need to understand what you need to do on that mRNA. If you need to modulate splicing in a specific location, you're going to put that ASO there in order to block that. If you want to affect the stability of the mRNA in determination, you go ahead and do that. So because of these two different strategies now of the ASO design, you can clearly see the different mechanism of action. The very first mechanism of action is the RNA degradation that I just went through with the gapmer, with the RNase H1. You have another one of a more promiscuous, if you like, enzyme, which is AGO2, which I'm not going to go through it at the moment. And then the second one is essentially you have mRNA modulation. You can reduce an mRNA, not necessarily by cleavage by RNase H1. You can have nonsense-mediated decay, no-go decay, and other things, which I'm not going to in the moment. So really, this shows how highly versatile and specific ASOs are that allow us and enable us to do what we do. When we understand what we need to do in this mRNA, we find the right class and we design the right class of the ASO in order to do that. Right, we have established that. Now we know we need to have a gapmer, we need to have a steric blocker, we know the chemical modifications that we need to do. How do we actually get these ASOs in our hands? How do we design these ASOs? So for that, and I'm very proud to say, we have our own in-house algorithm at n-Lorem. So hence the bioinformatic capabilities that I was talking about a moment ago. So we have an AI-informed ASO design, which actually makes the discovery extremely efficient. And what goes into that algorithm is what Stan was saying, right? Everybody talks about AI. You have an algorithm. The question is, what kind of data do you put into the algorithm for the algorithm actually to work good in a good manner? We are putting 35 years of ASO expertise and millions of ASOs. We are putting all relevant cell and animal models, hundreds and thousands of patients, and also our continuous learnings from whole genome sequencing. The whole genome sequencing data are the ones that really allow us to make this allele selective ASO. So we're putting all of this data together in order to perhaps be able to reach more patients with the same ASO, right in the same case of KEF1A. That you heard yesterday, because it targets the same benign SNP. And of course, we also use all of the state-of-the-art integration of multiomics data. Everything goes in there, all our expertise, and also internal capabilities, and then we end up with the ASOs. This is then moving to the discovery, the step 3. This is then we have the ASOs, we have synthesized them, we have them in the lab, and we start screening. What does that look like? The very first part of this, the real very first part, because there is a lot of optimization and validation that goes before that I'm not going to go into details today. But the very first important part is the primary ASO screen. And here, and actually this data is not a cartoon, this is real data that they have been screened in the lab from a patient of n-Lorem. And essentially what we do is we have about 500 ASOs that we have designed, and we screen them in patient cells. This graph might seem a little bit overwhelming. It's actually very simple. So what we are looking for here, and this is an allele selective ASO, we are measuring mRNA. We are measuring both the abnormal, the pathological mRNA, and the normal one. The normal is in blue. So because it's an allele selective, you would imagine that you don't want to reduce it. You want to keep it. But you want to reduce the red. which is the pathogen, with the abnormal, the mutant one. And you can already start seeing here that you have some spots that they're really, really good. You're seeing this blue up, red down. So these are essentially the ones, and as Dan said yesterday, we have very specific thresholds, and we have very specific minimum criteria. For the primary screen, efficacy is something that is very important. We only go for something that reduces more than 80%, as you can see there, of the mRNA target. So you see some ASOs there that they're super, super promising. So we are taking these, and then we move on to the real deal, dose response assay. So we usually take about 50 to 75 from the primary screen that we are seeing promising, and we scrutinize them. How do we scrutinize them? We basically do doses. So essentially we have and we create dose response curves, which essentially we just really checking these ASOs, putting them, okay, if you're a real deal, what should happen is you should be reducing the abnormal one as the dose goes up. So you should really see a dose-dependent reduction of the mutant. And this is essentially how we also, and by the IC50, we also measure potency. So here what we're looking for in the dose response are 3 criteria. We need the potency to be very good of the mutant, so we need an IC50 of less than 1 micromolar, very good efficacy, again, more than 80% of target reduction. And for an allele selective ASO, we also need a very good selectivity, so more than 10-fold. And you can see here these 3 guys at the very end that really meet this criteria. So these were some of the ones that then would move into in vivo tolerability studies. And from this step, we essentially usually take 20 to move them into in vivo tolerability studies. But before that, there are 2 additional steps that we do. The very first is also an in vitro off-target analysis. So we essentially want to confirm the selectivity. We want to confirm that the ASOs really act where they're supposed to act on their target without anything else. And lastly, of course, we're not going to take into animals any ASOs that we're already seeing in vitro are immunogenic. And for that, we have what we call the BJAB assay, where essentially scrutinize if there is any induction in the immune activation. So we only take ones that don't. So, and here again, I have 2 photos of the robots, and this is, where they really enable us to do all of this step in actually a very high throughput, rapid, rigorous, and cost-effective manner. Right, we also have very important decisions, critical decisions and challenges that we need to address. So the most important decision that we need to face is, at the end of this process, have we found the best ASO possible? If the answer is no, and we actually cannot go back because we have extended any possibility, we have to discontinue the case. But before that, I'll just show you in an example in a moment what kind of rationale we follow in order to do that, because we, of course, do not want to take this decision. So some of the challenges. We sometimes encounter differences with published data, and we really need to sort out what is the truth, let's say, because this is very crucial for our ASO design and discovery. We need the highest quality possible. Sometimes we don't know enough about the nature of mutation, so we actually do not know if the ASO and which kind of ASO is the best approach. Sometimes we encounter RNAs that they are very highly structured. You can imagine the way the ASOs bind. You don't want an RNA that is very highly structured. This is going to impede how the ASOs bind. So we need to find other ways to make this work. The last part is for allele selective ASOs. Remember I was telling you that we read the gene and we need to find spots that they have differences between the normal and the abnormal. Well, what if you don't have that many SNPs? What if you have a small number of these differences? Then what do? This is, and I will take you through a basic program that we are trying to eliminate these problems. Now, we really do everything that we can before we discontinue a case, because as I said, this is something that we do not want to do. The question that we ask when we reach that point is, are the ASOs sufficient given the function of the gene? So if we don't know that, we actually go ahead and take that ASO that we think is our optimal one, and we do research experiments. We test how a cell tolerates the specific ASO. Does it still do its normal function? Can the ASO rescue? If the answer, and we do in-house all of these research experiments, If the answer in all of this is no, then this means we have not found the optimal ASO. We cannot move forward with that. And if allele selectivity is not sufficient, meaning that we are also reducing the normal mRNA, or we're not reducing the abnormal enough, then if the answer to all of this is no, then we bring the case in and we recommend at the RMC to discontinue. This is an example to show you of some discrepancies that we are encountering with the published data. So for essentially we had a splicing case that had an indel mutation that was affecting 4 of our patients. According to published data, and you can actually see the highlights there, you can see that for one patient we didn't really have any information, and for the other 3, it seemed like the sequence was not the same. We needed to know this because actually without this, we were not able to go ahead and design these ASOs. So what we decided to do was actually do our own analysis. So we went ahead and we did long-read whole genome sequencing, and you can clearly see that actually all these patients are exactly the same sequence. And this was revolutionary. This is a aha moment, because this actually means that we can really design the same ASOs for all 4 patients, meaning with one ASO, we can treat 4 people. So a small discrepancy in one little base actually means a lot for us. And so we needed to find this answer. Now at the final part of my talk, I would like to switch gears and just tell, well, actually not switching too much. It's all research, and it all goes for the ultimate goal of finding, the best ASOs possible. But I will talk about directed research programs. So as I said, these are programs that we do when we do not know enough about the nature of mutation and the targets, and we need some further research before we even go ahead and start ASO design and discovery. And I will use as an example GIP3, JNK interacting protein. This is a very important protein, molecular scaffold, motor protein, very important functions in apoptosis, a neuron function, and all of that. So essentially, we had this particular mutation that we did not know. There was not enough information whether it was really a toxic gain of function. And the reason that was really crucial for us to understand is because a toxic gain of function mutation, you essentially have really that abnormal mRNA that creates a protein that takes over everything, does something bad. So really, an allele selective approach is perfect. This would be a very good therapeutic intervention, but we need to be sure that this is what is really happening. We also didn't know about the proximal molecular and pathological mechanism. So even if we got an ASO, we didn't even know what would rescue, what would be looking for. And then how much reduction of the wild type was tolerated. Allele selective, we need to know how stringent do we need to be. And this is important because you can imagine you can have more shots on goal if some of the reduction of the wild type is tolerated. And finally, can we actually ASO rescue? Is the ASO the optimal therapeutic strategy for this patient? We went ahead and answered all of this, and this is what we did research in-house. So now I'm very happy to say that yes, we have really established and determined that this mutation is a toxic gain of function, meaning that we could then go ahead and design and discover an allele selective ASO. We now know the proximal molecular pathological mechanism, and these include apoptosis, endosome mobility, and very importantly, a very novel function that nobody has reported, which is dopamine signaling. It disrupts dopamine signaling. And now you're going to say, okay, well, the impact of that is actually, it means that a proportion of Parkinson, juvenile Parkinson, it could actually be a GIP3 disease. And this is very important because that means that therapeutically, you can also use dopaminergic agents in combination with ASOs to actually treat these patients. Moderate reduction is tolerated on the wild type, which means that actually we had more shots on goal. So this is how then we knew our threshold. We knew what our thresholds were on the discovery process. And essentially, this is exactly what we did. We have defined a beautiful allele selective ASO. And yes, allele selective ASOs gave us a lot of confidence because really they rescued all the molecular pathological phenotypes, including the dopamine signaling I told you before. And now as I go towards the end of my talk, I'll just like to take a couple of moments to go for the basic research. Why do we do basic research? We want to advance the technology to really reach more patients today. 3 of these examples, and I will just take, for the interest of time, how can we expand to reach more patients? What do we need to do? What do we want to do? We want to have more efficient ASOs, safer ASOs, and reach a bigger range of patients. The very first thing that, of course, we're working on is we're working on mechanisms to avoid adverse events and immune activation. Why are we doing that? Because we want to make even better ASOs and even more efficient. Enhance allele selectivity. I told you throughout the talk that, with toxic gain of functions, allele selective ASOs are optimal, but we are limited sometimes by the number of SNPs. So 50% of our programs are allele selective, so we are really trying to find ways to enhance the allele selectivity. And eventually, of course, help even more patients rapidly. And finally, upregulation strategies. And this is very important. About 40% of our declined cases require an ASO-mediated protein upregulation strategy. And we are really working hard with promising results so far, but small steps. This is a huge effort. This is going to take a while to really be able to accept and treat these patients. And we have the knowledge and expertise to really address these issues today. As I conclude through the talk, I just wanted to take a take-home message. We have an outstanding team in process to discover optimal ASOs, and these are all their faces there. We have the capability to do this rapidly in a cost-effective and high-throughput manner, and really, we are 100% committed to you, to this community. The most important decision that we wake up every single day and we are making, every step of the way, is this ASO sufficient and optimal for this particular patient? And with that, I would really like to thank all of you. Thank you.